What if you could cut disease out of your DNA, like deleting a typo? No surgery, no chemo, no guesswork. Just precision editing at the genetic level, removing the cause of disease before it even starts? That's the promise of CRISPR. What began as a weird bacterial defense trick has exploded into one of the most powerful tools in modern medicine. And now, CRISPR Therapeutics is turning this science into real, life-saving treatments. From cancer to sickle cell to HIV, this isn't the future, it's happening now. What is CRISPR and how does it work? CRISPR isn't an abstract sci-fi concept, it's a molecular scalpel, precise and powerful. Short for clustered, regularly interspaced, short palindromic repeats, CRISPR was first discovered in bacteria, where it serves as part of an ancient immune defense system. These microbes use CRISPR to recognize and chop up viral DNA, essentially keeping a mugshot of past invaders to spot them if they ever return. Fast forward to the 21st century, and scientists realized they could hijack this natural defense tool. With a bit of bioengineering, CRISPR, paired with the Cas9 enzyme, became one of the most revolutionary tools in modern genetics. In this human-modified form, CRISPR, Cas9, can be used to edit DNA in living organisms with astonishing precision. Here's how it works. A specially designed piece of RNA, called the guide RNA, searches through the genome, like a molecular GPS. Once it finds its exact target, a specific DNA sequence, it directs Cas9 to that location. Cas9 then cuts the DNA at that spot. This opens the door for all kinds of genetic edits. Scientists can remove harmful mutations, insert healthy versions of genes, or even switch off problematic ones entirely. It's a simple idea on the surface, but the implications are enormous. From curing genetic diseases to creating virus-resistant crops, CRISPR is the beginning of a genetic revolution. From lab bench to hospital bed, the rise of CRISPR. CRISPR Therapeutics, the biotech company co-founded by Nobel Prize-winning scientist Emmanuel Charpentier, isn't just riding the gene editing wave, they're building the future of medicine. While CRISPR began as a revolutionary discovery in a petri dish, CRISPR Therapeutics is taking it far beyond the lab. Their mission? To develop gene editing therapies that don't just manage symptoms, but go after the root cause of disease and erase it entirely. This isn't theoretical anymore. It's happening in clinics, trial rooms, and hospital beds. CRISPR Therapeutics is tackling some of the most stubborn and deadly conditions known to medicine. Their trials focus on genetic blood disorders like sickle cell anemia and beta thalassemia, diseases that once meant a lifetime of pain, transfusions, and early death. With CRISPR, patients are being treated with a one-time gene editing therapy that reprograms their own cells. But they're not stopping there. The company is also targeting aggressive cancers like leukemia using modified immune cells that are trained to hunt and destroy tumors. And in the longer-term pipeline, treatments for chronic viral infections like HIV, conditions that have evaded cures for decades. And the trials? They aren't just promising, they're showing real, measurable success. For some patients, CRISPR has turned life sentences into fresh starts. The sickle cell breakthrough. Let's begin with a disease that has caused generations of suffering, sickle cell anemia. It's a brutal genetic disorder that twists red blood cells into rigid, crescent, or sickle shapes. These deformed cells don't flow smoothly through blood vessels. Instead, they clump, block circulation, and break down prematurely. The results are devastating: strokes, organ damage, and tragically shortened lives. But today, after decades of little progress, CRISPR is rewriting the script. CRISPR Therapeutics, in collaboration with Vertex Pharmaceuticals, developed a groundbreaking treatment called Exa-cel, short for exagam-glow gene autotem cell, and formerly CTX001. It's not just a new drug, it's a new approach to healing. A gene editing therapy tailored to the patient's own biology. Here's how it works. First, doctors extract bone marrow stem cells from the patient. In the lab, scientists use CRISPR to precisely edit those cells, switching on a powerful gene, normally active only in newborns, the fetal hemoglobin gene. This form of hemoglobin doesn't sickle, and it can take over the job of the faulty adult version. Once edited, the cells are reinfused into the patient, where they begin producing healthy, flexible red blood cells. For many patients, symptoms vanish. No more blood transfusions, no more ER visits. Some haven't had a single crisis since treatment. In clinical terms, it's a functional cure. In human terms, it's a second chance at life. Precision medicine at scale. It's not just another treatment, it's a fundamentally new kind of medicine. CRISPR doesn't mask symptoms, it doesn't slow the progression, it goes straight to the source and makes a correction at the root level. This is what sets it apart from traditional therapies. It doesn't manage the disease, it aims to erase it. The therapies aren't one size fits all. They're designed for you, for your unique genetic mutation, your personal code. This is what scientists mean when they talk about precision medicine. The ability to design treatments that work for a specific patient, not just general population. In cancer, it means engineering your own immune cells to recognize and kill only your tumor. In rare genetic diseases, it means repairing the one mutation that affects you and no one else. Even in brain disorders like Huntington's or ALS, there's growing hope that targeted gene edits could eventually halt degeneration. And here's the exciting part. As genetic sequencing becomes faster and more affordable, we're getting better at identifying exactly who needs what. That means fewer side effects, more effective results, and potentially the end of trial and error medicine. The future of healthcare is no longer reactive, it's personal. CRISPR versus cancer. Cancer isn't just one disease, it's hundreds of them. But at their core, all cancers share one thing. They're driven by genetic mutations. That makes the innovation a uniquely powerful weapon. Rather than using broad, often toxic chemotherapy, scientists can now go molecular, targeting the very mutations that cause cancer in the first place. CRISPR Therapeutics is leading the charge with a revolutionary approach, CAR T-cell therapy. In this process, a patient's own immune cells, called T cells, are transformed into cancer killers. Think of them as the body's natural defense force. But they're upgraded into something more: programmable, precision-guided assassins. Here's the process. Doctors first extract T cells from the patient's blood. Then, in the lab, CRISPR is used to rewire those cells in 3 key ways. First, the cells are equipped with a receptor that helps them recognize and bind to cancer cells. Second, they're edited to delete off switches, genes that would normally dampen their activity. And third, they're modified to evade rejection by the patient's body. Once reinfused, these edited T cells circulate through the patient's body, seeking out cancer and destroying it. In early clinical trials, patients with aggressive leukemias and lymphomas experience something once thought impossible: complete remission. This isn't just managing cancer. This is gene-edited warfare, and in many cases, it's working. Multiplex editing is the future. If editing a single gene can transform medicine, imagine what happens when we start editing several at once. That's the power of multiplex editing, the next leap forward in CRISPR's evolution. Instead of making just one tweak to a strand of DNA, scientists are now performing multiple edits simultaneously in the same cell. Take cancer immunotherapy, for example. A single edit might help immune cells recognize a tumor, but with multiplex editing, you can stack edits, giving those cells the ability to detect cancer, resist immune suppression, and avoid being rejected by the patient's body, all in one sophisticated genetic reprogramming. This opens doors far beyond oncology. Multiplex is being explored to treat autoimmune disorders, where multiple genes are misfiring at once. It's being tested in neurodegenerative diseases, where layered genetic pathways unravel over time. And some researchers are even looking at multiplex editing as a potential strategy for delaying aging, tweaking several longevity-related genes in one go. The takeaway? This innovation is becoming a platform, one capable of scaling up, layering complexity, and tackling the toughest biological puzzles we've ever faced. The future of gene editing is multidimensional, and it's arriving fast. CRISPR versus HIV, a viral challenge. HIV has been one of the most elusive and devastating viruses in modern medical history. For decades, we've had treatments that can suppress it but not cure it. That's because HIV is a master of disguise. Once it infects a person, it inserts its genetic material directly into the host's DNA, hiding in the very code of human cells. That's what makes it so hard to eliminate. But CRISPR might finally give us the upper hand. Instead of targeting the virus when it's active in the bloodstream, researchers are going after its hidden form, the integrated viral DNA, buried inside infected cells. Scientists can now locate this embedded sequence and cut it out with surgical precision. Imagine it like malware, lodged deep in your computer's operating system. CRISPR acts as the antivirus that finds and scrubs it clean. While the approach is still in its early stages, the results are promising. In animal models, researchers have already demonstrated complete viral elimination in certain tissues, a feat previously thought impossible. And now, the bold step. Human clinical trials are underway, testing this technology's ability to cure, not just control HIV. It's an audacious goal. But if the innovation can crack this viral puzzle, it would mark one of the greatest breakthroughs in the history of infectious disease. Gene editing and the brain, the next frontier. When we think of gene editing, we often imagine curing physical diseases, blood disorders, cancer, viral infections. But some of the most complex and heartbreaking conditions we face are rooted in the brain: Alzheimer's, Parkinson's, and Huntington's. These aren't just neurological disorders, they're genetic ones too. Each has a molecular trigger, mutations, or misfolded proteins that set off a cascade of damage. If we could intercept those signals at the source, we might be able to stop these diseases before they cause irreversible harm. That's where this innovation comes in. But there's a catch: the blood-brain barrier. It's one of the body's most formidable defenses. A tightly sealed border that blocks most substances, especially large molecules, from entering the brain. Delivering gene editing tools to this protected environment is one of the biggest challenges in medicine. To overcome it, scientists are designing Trojan horses, ingenious delivery systems like viral vectors and nanoparticles that can carry past the barrier and into brain tissue. Some of these are disguised as molecules the brain welcomes in, sneaking the payload across undetected. If they succeed, we could edit brain cells to prevent neuron death, reduce inflammation, or correct mutations before they cause symptoms. It could transform how we approach memory loss, motor decline, psychiatric illness, and more. Gene editing in the brain isn't just a possibility, it's the next great leap. Gene therapy meets regenerative medicine. This isn't just about deleting what's broken, it's about building what's missing. In the world of regenerative medicine, gene editing is joining forces with stem cell science to push biology into new territory, growing tissues and organs from scratch. Here's how it works. Scientists first take ordinary adult cells, like skin or blood cells, and reprogram them back into stem cells, the raw, flexible building blocks of the body. Then, using CRISPR, they edit out any harmful mutations, ensuring those cells are healthy and genetically sound. Finally, under the right conditions, those edited cells are coaxed into becoming specific types of tissue: heart, liver, skin, even neurons. Scientists have used CRISPR-corrected cells to regrow skin for burn victims, creating sheets of healthy tissue that integrate seamlessly with the body. Other teams are developing patches of functioning heart tissue for patients who've suffered heart attacks. But the long-term vision goes even further. Custom-grown, gene-edited organs that are biologically matched to a patient, no risk of rejection, no need for lifelong immunosuppressants. Imagine a future where a failing liver isn't treated or replaced, it's regrown. Where gene therapy and regenerative medicine don't just save lives, they rebuild them cell by cell. Risks, challenges, and ethical frontiers. CRISPR holds stunning promise. But let's not get carried away. This isn't a magic wand, it's a molecular scalpel. And like any powerful tool, it comes with risk and repercussions. First, the science itself isn't flawless. It can cause off-target effects, where the wrong section of DNA gets snipped or altered sometimes with dangerous consequences. Also, delivery still remains as a technical hurdle. How do we get into the right cells, in the right tissue, at the right time, without triggering an immune response? But perhaps the trickiest challenges are ethical. What happens when editing isn't just about curing disease, but enhancing traits? Designer babies aren't theoretical anymore. Editing embryos to select height, intellect, or eye color may soon become possible, and deeply divisive. Then there's germline editing, changes that get passed down to future generations. If something goes wrong, it's not just one person affected, it's their children, and their children's children. And what about access? Will these therapies be available to everyone or just the wealthy? Will the innovation deepen global health inequality? Do we trust governments? Probably not. Corporations? Who sets the boundaries for what genes are defective and what counts as an improvement? These aren't questions science can answer on its own. They demand a broader conversation across cultures, laws, and values. Because editing genes is one thing. Editing humanity? That's another. Regulation and the road to approval. Turning a CRISPR-based therapy into an approved medical treatment means navigating some of the strictest oversight in modern medicine. Every candidate drug must prove it's not only effective, but also safe, stable, and predictable across time and diverse populations. That means clearing multiple regulatory hurdles, including, but not limited to, long-term safety. Do the gene edits stay where they're supposed to? Edit stability. Will the changes remain in place as cells divide? Off-target risks. Are there any unintended mutations even years later? And therapeutic benefit. Does the cure genuinely outweigh the risks involved? Each stage takes years. And regulators from the FDA in the United States to the EMA in Europe are cautious, as they should be. We're not talking about managing symptoms here. We're rewriting DNA. As of 2024, CRISPR Therapeutics and Vertex Pharmaceuticals have already hit a major milestone. Exa-cel, their therapy for sickle cell disease, has been approved in the UK and Europe, with FDA approval, pending in the US. If it passes, it will become the first CRISPR-based therapy to go mainstream, a historic first in genetic medicine. But it won't be the last. Dozens more are already in the pipeline. The age of gene-edited medicine is just beginning. A platform, not just a treatment. Think about electricity or the internet. These weren't single-use technologies. They were foundational platforms that changed how we do everything, from communication to commerce to medicine. This breakthrough is on that level. It doesn't just fix one disease. It redefines what's possible across biology. At its core, the innovation is a programmable system. Change the guide RNA and you change the target. That means the same basic editing toolkit can be adapted to thousands of conditions, from rare genetic mutations to widespread illnesses like cancer, HIV, and Alzheimer's. It's modular, like Lego bricks for your DNA. It's scalable, capable of being deployed in cells, tissues, even entire organisms. And it's getting better, more precise, faster, safer, with every passing year. This is why it matters. Not just because it can treat sickle cell or target tumors, but because it reshapes the way we think about medicine, about health, and even about what it means to be human. We're not just patching up biology anymore. We're learning how to rewrite the source code of life itself, with intention, precision, and hopefully, wisdom. So here's the question. If you had the power to cut disease out of your genes, would you use it? The innovation is still young, but it's growing fast, editing blood, immune cells, even the brain. It could cure diseases we've battled for centuries, but it could also change what it means to be human. If you want to stay ahead of the revolution in biotech and human health, like this video, plus subscribe. Thank you for watching. Stay blessed and stay curious.